Hello and welcome to OSDR Chats. How can datasets from the Open Science Data Repository provide clues about health in spaceflight conditions? Today we have the opportunity to speak with Dr. Emmanuel Gonzalez from McGill University, whose team used 3 datasets from the OSDR to research how spaceflight-associated changes affect the gut microbiome's interaction with our bodies, as well as the implications of human health on long-duration space missions. In this interview, Dr. Gonzalez shares this research with us and describes how the Open Science Data Repository and the analysis working groups contributed to this publication. Thanks for joining us today, Dr. Gonzalez. Could you please highlight the most important finding in your study?
Yeah, sure. It's great to be here. And one of the key takeaway from our study was seeing a drop in beneficial gut bacteria like Ectibacter maiores and Disosmobacter welbionis in mice after flight. And these bacteria are very important for bile acid and butyrate metabolism, and their reduction seemed to be linked to the unique conditions of space, like microgravity and confinement. And what's striking is how these changes were tied to metabolic shifts in mice, including insulin resistance and increased liver stress. And maybe even more interesting, these effects in mice are similar metabolic and physiological changes that have been observed in human astronauts after space missions. So it's a powerful connection that shows just how much the microbiome can influence overall health in extreme environments like space. And I might just add one thing. We used a high-resolution bioinformatics approach, and that let us detect rare bacterial species across samples. And that gave us really a clear picture of how these systems are changing. And what really stood out is how shifts in gut bacteria were reflected in the gene expression of the host liver and colon, showing a tight link between microbiome and host physiology. And this kind of interplay really highlights some of the long-term health risks astronauts could face on future missions to Mars and beyond.
Thanks for sharing those insights with us. I have a second question. How did the Open Science Data Repository or the analysis working groups assist you in your work so you could achieve these research goals?
Yeah, well, what's great about analysis working groups and open data is how they bring together people from different fields, and that mix of expertise was key in helping us to look at the data from multiple angles and really make sense of what we were seeing. And really, this kind of collaboration makes open science so powerful. It pushes research further than any one team could do on their own. And one of my favorite parts of this project was working with Gene Lab for a high school program. We had 3 students, Angelamo, Alice Finkelstein, and Peyton Walton from the 2022 cohort. And those 3 students analyzed RNA-seq data, so transcriptomics, from the liver and colon. And their findings really lined up with our microbiome results. And they helped us to show that this complex relationship between the gut microbiome and the host physiology during space flights. And it was incredible to see high school students contributing to that level. And we were all authors. We were very proud to have them as co-authors.
I'm glad to really see the fruits of your collaboration. I'm really thankful that you were able to work with these high school students. So thank you for that. And to our audience out there, you can learn more about this work, how to join the analysis working groups, and access the datasets analyzing this work with the link provided in the description section below. Thank you and have a wonderful day.
Thank you.